In the previous video that I uploaded on dairy technology, I covered lactose and water as one of the components of milk. The next components of milk which I'm going to cover in this video are proteins and fats. I'll start with proteins. The building blocks of proteins are amino acids. Examples of amino acids are glycine and alanine. Milk contains around 30 to 36 grams per liter of proteins, hence milk has high nutritive value. It is believed that milk is a good source of protein because it contains all essential amino acids. I remember saying in one of my previous videos that amino acids are categorized into two groups, that is essential and non-essential amino acids. Remember, essential amino acids are amino acids that cannot be synthesized by the body, hence your body must obtain these amino acids from the food you consume. Milk proteins are categorized into two groups, that is casein proteins and whey proteins. Caseins comprises of 80% of milk proteins. They majorly contain covalently linked calcium phosphate groups. These groups make these caseins to interact with each other to form large spherical micelles. The diameter of these micelles vary from 30 to 300 nanometer. Casein proteins consist of alpha S1 casein, alpha S2 casein, beta casein, kappa casein, and gamma casein, while whey protein comprises of 20% of milk proteins. Whey proteins consist of alpha lactalbumin, serum albumin, beta lactoglobulin, and immunoglobulins. The difference between casein and whey proteins is observed when you add acid or rennet enzyme to milk. When acid or rennet enzyme is added to milk, casein micelles combine to form curds which are in solid form, while whey proteins remains in liquid form. This science is applied in cheese making, so I'll explain more of this when covering cheese making. Let's see how proteins are synthesized in the secretory cells of the mammary glands. I've said the building blocks of proteins are amino acids. Amino acids are supplied to secretory cells by the blood. Synthesis of protein starts in the nucleus of the cell. The nucleus has deoxyribonucleic acid, which contain the genetic instruction to build every protein in the body. The nucleus also has an enzyme called ribonucleic acid polymerase, whose function is to unwind section of DNA to expose genetic instruction needed to make milk proteins. After unwinding, the instruction is copied to form messenger ribonucleic acid. Messenger ribonucleic acid strand carries the genetic information for making proteins from the nucleus to the ribosomes. In the ribosomes, messenger ribonucleic acid combine with amino acids in a determined sequence to form milk proteins. Proteins formed are folded into particular shapes. Remember, proteins can be in spherical or round shapes. Folded proteins are taken to the lumen of the Golgi apparatus, where they are linked to groups like carbohydrates and phosphate groups. Casein micelles are also formed in the Golgi apparatus. Some proteins like immunoglobulins are not synthesized in the secretory cells but are obtained direct from animal blood. So after synthesis, the synthesized proteins as well as absorbed proteins pass through membrane to lumen of alveoli for storage. The next milk component is fats. Fats are made up of fatty acids and glycerol. Fatty acids are derived from the feed and supplied to secretory cells by blood and lymph. Milk fats are majorly made up of saturated fatty acids like palmitic and butyric fatty acids. Remember, saturated fatty acids are fatty acids that do not have double bond, while glycerol is synthesized from glucose. Fats are synthesized by the endoplasmic reticulum, where fatty acids are attached to glycerol. Fats formed are directed to the cytoplasm, where they collect as lipid droplets. These droplets move to the apical plasma membrane, where they associate to form large spherical globules. Globules vary in size from 2 to 6 micrometers. These globules are coated with a surface material which is made up of proteins and polar lipids like phospholipids and cholesterol. The function of the coat is to stabilize fats in aqueous phase of milk. The coated globules are expelled into alveoli lumen for storage.